Welcome to Shiney Brain Academy. Today we are studying carbohydrates, monosaccharides, disaccharides, polysaccharides, reactions in chemistry. This is a very important topic that every student must understand well. In this video lesson, we will cover all the key concepts, definitions, examples, and real-world applications related to carbohydrates, monosaccharides, disaccharides, polysaccharides, reactions. Follow along carefully, take notes, and by the end of this lesson you will have a thorough understanding of carbohydrates, monosaccharides, disaccharides, polysaccharides, reactions, and how it relates to chemistry. Let us get started right away. Carbohydrates are fundamental organic molecules essential for life. They are composed of carbon, hydrogen, and oxygen atoms, often in a ratio of CH2O. Think of them as the fuel for our bodies and the building blocks for many structures. In our diet, carbohydrates are a crucial energy source, powering everything from our brain to our muscles. Beyond energy, they play vital structural roles, like in the cell walls of plants and the exoskeletons of insects, showcasing their diverse importance in the biological world. A key characteristic of many carbohydrates is their general formula, which is CH2ON. Here, N represents a whole number, typically 3 or greater. This formula highlights the characteristic 2:1 ratio of hydrogen to oxygen atoms, similar to that found in water. While this formula is a useful starting point, it's important to remember that not all compounds fitting this formula are carbohydrates, and some complex carbohydrates have slightly different compositions, requiring a deeper structural analysis for classification. To understand the vast world of carbohydrates, chemists classify them based on their size and complexity. This hierarchical system simplifies their study and helps us grasp their diverse roles. The 3 main categories are monosaccharides, the simplest units, disaccharides, formed from 2 monosaccharide units, and polysaccharides, which are large polymers composed of many monosaccharide units. Each of these classifications possesses unique chemical and physical properties, leading to a wide array of functions within living organisms, from immediate energy to long-term storage and structural support. Let's begin with the most basic building blocks, monosaccharides. These are simple sugars, meaning they cannot be broken down into simpler carbohydrates through hydrolysis. They are the fundamental units from which all larger carbohydrates are constructed. Common examples include glucose, the primary sugar used by our cells for energy, fructose, often called fruit sugar, and galactose, a component of milk sugar. Their simple structures allow them to be readily absorbed and utilized by the body for immediate energy needs. Glucose is arguably the most important monosaccharide. It's an aldohexose, meaning it's a 6-carbon sugar with an aldehyde functional group. Glucose is the primary sugar circulating in our bloodstream, serving as the main fuel source for our cells. Its production is a remarkable feat of nature, occurring during photosynthesis in plants, where light energy is converted into chemical energy stored within glucose molecules. This makes glucose the starting point for most energy pathways in aerobic organisms. Next up is fructose, also a 6-carbon sugar, but it's classified as a ketohexose because it contains a ketone functional group. Fructose is famously known as fruit sugar due to its high concentration in ripe fruits. It's also found in honey and is a component of sucrose. A key characteristic of fructose is its intense sweetness, making it considerably sweeter than glucose. This property contributes to its use as a sweetener in various food products. Galactose is another important aldohexose, sharing the same chemical formula as glucose and fructose but differing in its structure. While it exists as a monosaccharide, its most common occurrence in nature is as a component of lactose, commonly known as milk sugar. Lactose is a disaccharide found in mammalian milk. Galactose is not as sweet as glucose and plays a crucial role in the metabolism of newborns and in the formation of certain biological molecules. Moving up in complexity, we encounter disaccharides. These molecules are formed when 2 monosaccharide units are covalently linked together. This linkage occurs through a special bond called a glycosidic bond, formed via a dehydration reaction, where a molecule of water is removed. Common examples of disaccharides include sucrose, commonly known as table sugar, lactose, the sugar found in milk, and maltose, often called malt sugar. These are larger than monosaccharides but still soluble and digestible. Sucrose, the familiar table sugar we use in our kitchens, is a prime example of a disaccharide. It's formed by the combination of 1 molecule of glucose and 1 molecule of fructose, joined by a glycosidic bond. This bond is specifically an alpha-1,2 glycosidic linkage. Sucrose is extracted from sugarcane and sugar beets and is the most widely consumed and recognized sweetener globally, playing a significant role in our food industry and culinary practices. Lactose, often referred to as milk sugar, is another important disaccharide. It's formed from the union of glucose and galactose, linked by a beta-1 for glycosidic bond. Lactose is the primary carbohydrate found in milk produced by mammals, providing a vital energy source for infants. Many individuals, however, experience lactose intolerance, meaning they lack sufficient lactase enzyme to break down lactose, leading to digestive discomfort. Understanding lactose is crucial for those with dietary sensitivities. Maltose, also known as malt sugar, is a disaccharide formed from two glucose units linked together. The specific linkage is an alpha-1 for glycosidic bond. Maltose is a common product of starch digestion, and it's particularly prevalent in germinating grains like barley, where it's used as an energy source for the growing seedling. It also plays a role in the brewing industry and is found in some syrups and malted beverages. Finally, we arrive at polysaccharides, the largest and most complex class of carbohydrates. These are essentially long polymers, or chains, made up of hundreds or even thousands of monosaccharide units, typically glucose, linked together by glycosidic bonds. Because of their complex structure, they are often referred to as complex carbohydrates. Prominent examples include starch, the primary energy storage form in plants, glycogen, the storage form in animals, and cellulose, a crucial structural component of plant cell walls. Starch is the principal way plants store energy. It's a polysaccharide composed of glucose units, but it has two distinct forms, amylose, which is a linear chain, and amylopectin, which is a branched chain. The ratio of these two components can vary. For humans and many other animals, starch is a vital dietary source of glucose. Our digestive system breaks down starch into glucose, which is then absorbed into the bloodstream to provide energy for our cells and bodily functions. Glycogen serves as the primary storage form of glucose in animals, including humans. It is predominantly stored in the liver and muscles. Structurally, glycogen is similar to amylopectin in starch, being a highly branched polysaccharide of glucose units. This extensive branching is crucial as it allows for rapid release of glucose when the body needs a quick energy boost, making it readily available for cellular respiration and maintaining blood glucose levels. Cellulose is a structural polysaccharide found in the cell walls of plants, providing rigidity and support. It's composed of beta-glucose units linked by beta-1 for glycosidic bonds. This different linkage compared to starch and glycogen is key. Unlike starch, humans cannot digest cellulose because we lack the enzyme, cellulase, required to break down these beta-1 for glycosidic bonds. This indigestible fiber is crucial for digestive health, acting as dietary fiber and aiding in waste elimination. Chitin is another significant structural polysaccharide, though it differs from cellulose by containing nitrogen. It is a primary component of the exoskeletons of arthropods, such as insects and crustaceans, providing them with their hard, protective outer shell. Chitin is also found in the cell walls of fungi, contributing to their structural integrity. Its unique composition makes it a valuable material in various applications, from biodegradable packaging to medical sutures. The formation of a glycosidic bond is a fundamental chemical reaction in carbohydrate chemistry. It's the process by which monosaccharide units are joined to form disaccharides and polysaccharides. This linkage is created through a dehydration reaction, also known as a condensation reaction. In this process, a molecule of water is removed from the two reacting monosaccharides, allowing them to form a covalent bond. The specific type of glycosidic bond, e.g., alpha or beta, and the carbon numbers involved, determines the properties of the resulting carbohydrate. In contrast to formation, hydrolysis is the process by which complex carbohydrates are broken down into simpler ones. This reaction involves the addition of a water molecule to the glycosidic bond, effectively breaking it and separating the monosaccharide units. This process is crucial for digestion, where enzymes like amylase, sucrase, and lactase act as catalysts, speeding up the hydrolysis of starch, sucrose, and lactose, respectively, into absorbable monosaccharides. A key chemical reaction of carbohydrates is oxidation. Monosaccharides that possess a free aldehyde, or aldose, or a free ketone, ketose, group can undergo oxidation. Such sugars are known as reducing sugars because they can reduce other compounds, such as metal ions in Benedict's or Fehling's solutions, while they themselves are oxidized. Glucose and fructose are excellent examples of reducing sugars. This property is often exploited in chemical tests to detect the presence of these simple sugars. Carbohydrates, with their abundant hydroxyl, OH, groups, readily undergo esterification. This reaction involves the interaction of these hydroxyl groups with acids, leading to the formation of ester linkages. Esterification can significantly alter the properties of carbohydrates, affecting their solubility, reactivity, and even biological function. For instance, the phosphorylation of glucose in metabolic pathways involves the formation of phosphate esters, which is critical for energy transfer and regulation within cells. Now let us test your understanding. Here is your question. Which of the following is a polysaccharide that serves as the primary energy storage molecule in animals and is highly branched? A. Cellulose. B. Starch. C. Glycogen. D. Sucrose. Pause the video, think carefully, and select your answer. That is all for today's lesson on carbohydrates, monosaccharides, disaccharides, polysaccharides, reactions. We hope you found this video clear and helpful. If you have any questions, join our Telegram channel at t.me/ShineyBrainAcademy, where our teachers are always available to help you. Do not forget to share this video with your classmates and friends who are also studying chemistry. See you in the next lesson. Goodbye and keep learning.